Welcome to all students. Today we are going to study about the development of the anther as well as microsporogenesis. First we will be studying how the development of the anther occur, and then we'll be studying how do microsporogenesis occur. Okay. Now very first you must know that the development of the anther in the advanced angiosperm is of eusporangiate type. Say for if I talk about the capsula, which is in typical angiosperm, then in capsula the development of anther is eusporangiate type. What is eusporangiate type of development? Development of the anther from more than one archisporial cell. Development of the anther from more than one archisporial cell is called as eusporangiate development. There is one more type of the development called as leptosporangiate type of development, in which the anther develops from a single archisporial cell. But here the development of the anther occur from more than one archisporial cell, and hence it is called as eusporangiate type of development. Okay. Now if we want to study the development of the anther, then we have to view these three diagrams, diagram number A, B and C. In the very first diagram, you are able to see that anther is a spherical or a oval structure. Anther is a spherical or a oval structure which is having a mass of meristematic cells, which is having a mass of undifferentiated meristematic cells, and it is surrounded by the epidermis, You are able to see the diagram number A. It is a mass of meristematic cells surrounded by the epidermis. So what I want to say, that anther appears as an outgrowth like structure. Initially anther appears as an outgrowth like structure which shows spherical or oval structure. Means initially when the development of anther starts, it is just like an outgrowth, and it seems to be spherical or a oval structure. Fine. And at this stage it is a mass of the meristematic cell. Means at this stage it is having undifferentiated meristematic cells, and these meristematic cells are surrounded by a single layer, a single thick layer, which is called as the epidermis. Is it clear? And very first you must know that during the development of the anther, at the middle portion, vascular tissues are developed. Vascular tissues are formed or developed. Okay. Now just view the diagram number 2. Now you must know that what is hypodermal region. The region present below the epidermis is hypodermal region. So you have to concentrate on the region just below the epidermis. Fine. Now simultaneously group of cells located just below the epidermis, in vertical rows of hypodermal region. What I have said just now, the region present below the epidermis is the hypodermal region. So what happens? Simultaneously group of cells located just below the epidermis in the hypodermal region at the four corners become visible with large nucleus and dense cytoplasm. Means what happens, that the region just below the epidermis is what? The hypodermal region. So in this hypodermal region at the four corners, 1, 2, 3 and 4, because we must know that at the maturity the anther seems to be a tetragonal structure. Okay. So at the four corners become large with visible nucleus and dense cytoplasm, due to which they are different from rest of the cells, and these are called as archisporial cells. Are you getting? So what I want to say, that group of the cells which are located just below the epidermis, that is in the hypodermal region, at the four corners become large, which are having a visible clear-cut nucleus and dense cytoplasm, right? And because of which these cells seems to be different from the other cells. These group of cells present just below the epidermis in the hypodermal region at the four corners of the anther are called as archisporial cell. So are you able to see the archisporial cells? And these archisporial cells are having a clear-cut visible nucleus and dense cytoplasm, and because of which these cells are different from the other cells. Okay, now in the next diagram, that is the diagram number C, now what happens? These cells means the archisporial cells which are present at the corners in the hypodermal region divides periclinally into two cells. Say for this archisporial cell divides, this archisporial cell also divides, all the archisporial cells divides. So these cells divide periclinally into two cells: outer primary parietal cell, which I have written in short primary parietal cell PPC, and an inner PSC. PSC means primary sporogenous cell. Again I am repeating, archisporial cell divides into two cells: outer PPC, inner PSC, primary sporogenous cell. Now both these cells undergo division. Both these cells will undergo divisions to form the complete structure of the anther except the epidermis. Now remember this thing, epidermis is not at all formed by the archisporial cell. This I have said earlier also. If you have seen my video, you know very well that anther is surrounded by four layers: epidermis, endothecium, middle layers and tapetum. But always remember that epidermis is not at all formed by the archisporial cell. Endothecium, middle layers and tapetum will be definitely formed by the archisporial cell, but epidermis is not formed. So what I want to say, that the archisporial cell now divides periclinally into two cells: outer PPC and inner PSC. Okay, and both these cells will form the complete structure of the anther except the epidermis. Fine. Now let's talk about the outer cell known as the primary parietal cell. What does this PPC cell forms in anther? So PPC cell, primary that is parietal cell, undergo periclinal division as well as anticlinal division. It undergoes periclinal division as well as anticlinal division. What are periclinal division? Divisions which occur parallel to the cell surface. And what are anticlinal division? Divisions which occur perpendicular to the cell surface. So the PPC cell, the outer cell, divides by periclinal division and anticlinal division to form the wall layers around the anther. And which wall layers? Endothecium, middle layers and tapetum. What is endothecium? What is middle layer? What is tapetum? When I have talked about in my video about the structure of the anther, I have given the description of this endothecium, middle layers and tapetum. If you have not watched up till that video, you go and watch that video. Now, what about the second cell? PSC. PSC is the cell present below the PPC towards the center. So primary sporogenous cell divides by mitotic division to form the sporogenous cells. Now we know very well that group of sporogenous cell is also called as sporogenous tissue. So don't be confused. Group of sporogenous cell is called as sporogenous tissue, which later gets differentiated. Means the sporogenous cells later gets differentiated into microspore mother cell. Microspore mother cell is also called as PMC, pollen mother cell. Why? Because these microspore mother cells or the pollen mother cells will form later the microspores or the pollen grains. Fine. So in nutshell, I can say that the primary parietal cell forms the wall layers around the anther, and primary sporogenous cells form the primary sporogenous cell form sporogenous cell, which later differentiates into microspore mother cell, and microspore mother cell will form what? The microspore. Now the question arises that this is the development of the anther, but along with the development of the anther, we are also studying the microsporogenesis. And what is microsporogenesis? Microsporogenesis is the formation of the microspore, from the microspore mother cell. So each microspore mother cell, remember this thing, microspore mother cell is a diploid cell. Each microspore mother cell divides by meiosis. We know very well that meiosis is a reductional division and will reduce the chromosome number to half. So if microspore mother cell is diploid, so definitely by meiosis the microspores which are formed will be haploid. So always remember, development of the microspore from microspore mother cell is called as microsporogenesis. When one microspore mother cell divides by meiosis, at least how many microspores are formed? 4 microspores are formed, and all 4 microspores are functional. And one more thing, initially the 4 microspores formed by the division of microspore mother cell are attached together with the help of callose layer. Like this, if 4 are formed, they all are attached together with the help of callose layer. Okay, and this group of 4 microspore, is called as a tetrad. But after some time, the callose layer, because of which the 4 microspores were held together in a tetrad, what happens? This callose layer is dissolved by an enzyme known as a callase enzyme, which is secreted by the tapetum. Tapetum secretes callase enzyme dissolves callose, because of which all the 4 microspores found in the tetrad become separate. Fine. And in the previous videos you know that Ubish bodies are secreted by the tapetum. So Ubish bodies are secreted by the tapetum in the microsporangium or the pollen sac, and that Ubish bodies forms the exine layer. That Ubish body secreted by the tapetum forms exine around the microspore. Always remember, when microspores are surrounded by the exine made up of the sporopollenin, then that microspore is now called as what? The pollen grain, which is now called as pollen grain. Okay, so this is all about the development of the anther and the microsporogenesis. Now in the next video we will be dealing with that the tetrad which is formed by the microsporogenesis or by the division of the microspore mother cell. How many types of the tetrads are there? You might have heard about tetrahedral tetrad, isobilateral tetrad, okay, and 3 more types of the tetrad, linear tetrad and all. That we'll be discussing in the next video. So thanks a lot for watching me.